Assalamu alaikum, students.
Walaikum salam, doctor.
Okay, there are others still missing, maybe 38. Okay, we'll start. We cannot wait. Okay, so today we'll talk about the beginning of the life, that is the first week of development. So we'll talk about the fertilization, cleavage, as well as implantation. So these are the topics which we'll try to cover here. The first is we should know what exactly is fertilization. Okay, so the fertilization, as you know, is a process of fusion of two mature gametes, that is the germ cells. In case of male, these are called as the spermatozoon or sperm, and in case of female, it is called as an ovum. So when the fertilize the sperm and the ovum come and fuse together, then it is called as fertilization. So the sperm and the ovum, if you see, they are haploid gametes. Haploid means half the number of chromosomes. If you know, there are 23 pairs of chromosomes. But in case of the sperm and the ovum, there will be only half of the chromosomes. That is 23, not 23 pair, but 23. So when the 23 from the male sperm come and fuses with the 23 from the female, then this process is called as fertilization. So then it forms a single cell. Okay, and this fertilized egg will be called as the zygote, and it will be diploid number chromosome. Now it becomes 23 pairs. Initially there will be sperms will have 23 and ovum will have 23 chromosomes. When they fuse together, when they fertilize, then they become diploid number, means double the chromosome, that is the normal number of chromosomes, 23 pair. Okay, so that process of fusion of the male and the female germ cells, that is the spermatozoon and the ovum, to form a single cell, that is the zygote, is called as the fertilization. Okay, fertilization takes place in the ampulla. As we know, the fallopian tube is a long tube and it has four parts. One is infundibulum, then we have the dilated part called as ampulla, then we have the isthmus, narrow part, and the most narrow part, that is called as intramural part. Okay, out of this, fertilization usually occurs in the ampulla, most dilated part of the fallopian tube called as the ampulla, where the fertilization will take place. This fertilization is divided into three stages. It is divided in three stages. The first is approximation of gametes, means how the gametes, the male and the female gamete, will come close. Okay, that is called as approximation of the gametes. The second is contact and fusion of the gametes. Contact and fusion, how they come in contact and how there will be fusion of the gametes, how they fuse together. Then the third is what are the effects after fertilization takes place? What are the changes which we can see? Okay, so that we'll study. So this will study. So this fertilization will study in three stages. The first stage is the approximation of the gametes. Okay, so as you know, the sperms and the ova reach the ampulla. So the sperm will come all the way from the vagina through the cervix into the uterine cavity and floats all the way up to the site of fertilization, that is the ampulla, and the ovum will come from the ovary, enter the infundibulum and reach us here. Okay, so the sperm and ovum reach the ampulla. Deposition of the spermatozoon in vagina leads to prostaglandins of the semen, including the powerful contraction of the uterine muscles. So whenever there is deposition of these sperms in the vagina, it leads to the prostaglandin of the semen producing powerful contraction of the uterus. So even though the prostaglandin are coming from the male, so it leads to powerful contraction of the uterine muscles. So also the coital reflex releases oxytocin, leading to contraction of the uterus. So two things will combine together. One is the prostaglandin, which are present in the semen, will help in the contraction of the uterus. The second is the coital reflex itself will lead to the oxytocin release in the female, and it leads to contraction of the uterus. So the contraction of uterus is both by the male semen as well as the oxytocin in the female. Now, mixture of sperms with the secretion of uterine glands is passively aspirated. Now, the sperms which are the semen which is released into the vagina, now they are passively aspirated into the uterine cavity by the mixture of the sperms as well as the secretions of the uterine glands. Uterine glands are within the uterus. Then there will be active motility of the sperm does not help. The sperms can move on their own with the waddling of the tail. They waddle the tail and they can move. But here in this stage, the activity of the sperm doesn't help much. Okay, so if you see here, this is the view of this one, okay, where the tail will start moving, okay, and here also it will move in a continuous rounded direction. Actually, I'm presenting the PDF version. If not, I could have shown you the motions here. The PowerPoint motion has the motions here, you can see, but because this PDF, this cannot move. Okay, so the tail can move, it can show moving as well as it also winds around. This is how the tail will move, and that helps in the movement of the sperm. But here at this stage, it doesn't help much. The spermatozoa are gradually reduced in number by the barriers provided by the abrupt contraction of the cervix and the uterine ostium. So once here the sperms are poured inside, the contraction will begin, and this contraction will lead to production of many barriers which will prevent the sperms from entering. Okay, so out of the millions of sperms which are poured inside, out of the 200 to 300 million sperms emitted at a single ejaculation, only 1% enter the cervix. So from the vagina up to the cervix, so only 1% will reach the cervix. Okay, in the vagina, if there are 200 to 300 million, only 2 to 3 million can reach the cervix. So there is the first barrier which will prevent the sperms from moving. And most of the sperms die within 24 hours. So they are viable only for 12 to 24 hours, and most of the sperms will die within 24 hours. So from trip from the cervix to the oviduct, that is the fallopian tube, takes at least from 30 minutes to 6 days. The fastest can move from the cervix up to the ampulla part of the oviduct or the fallopian tube. It can be as fast as 30 minutes, and some can go and they can be some of them can be alive up to 6 days, and they can reach up to maximum 6 days. But most of the sperms will reach an average within 24 hours. Okay, so the fastest can reach within 30 minutes, and the very slow one can reach up to 6 days. The oocyte is transported by the ciliary beads and the rhythmic contraction of the musculature of the uterine tubes. The sperms, we know it can move with the tail waddling. Okay, but here, as I said, it has a very less effect. But the main contraction is by the contraction of the uterus, which will pull the as well as aspirate the sperms, and it will push into the fallopian tubes. But how do you the oocyte will move? Okay, so oocyte is transported by the ciliary beads and rhythmic contraction of the musculature of the uterine tubes. Within the uterine tubes, if you study, when you study the histology, you will see that the fallopian tubes have ciliary cilias within them, and they will beat towards the uterine cavity, and they will pull the oocyte from the ovary into the ampullary part. Okay, so this is how the oocyte will look, and these are the sperms. Okay, so now here you can see here, this is the oocyte here inside, and all this around this oocyte are the sperms. What you are seeing, the whitish structures here, all these are the sperms. One oocyte and surrounded by hundreds of the sperms. So now the second stage is contraction and fusion of the gametes. Okay, So out of the 200 to 300 million sperms which we saw which will be released with each ejaculation, only 1 or 2 to 3 million will reach the cervix, and out of that, only 300 to 500 sperms actually reach the ovum. So what sperms are covering this oocyte are only 300 to 500 sperms out of the millions of sperms. And out of this 300 to 500 sperms, only one is able to penetrate. Only one of them penetrates the oocyte. And what is the function then? Why so many sperms are there? So the rest are engaged in disintegration of the three barriers around the secondary oocyte by an enzyme called as the hyaluronidase. Hyaluronidase will be released by these other sperms. Only one can enter, and the rest will release the hyaluronidase, which will break the barrier. There are three barriers. We'll talk about that. So there are three barriers which are supposed to be broken down. Okay. So this hyaluronidase will be present in the acrosomal cap of the sperm. If you study the sperm has a head, and on the top of the head there is acrosomal cap. So this is what we are supposed to see in the seminar presentation, but the students have very less information. They don't give any information. Okay. Okay. So they are supposed to tell the whole structure. How is the acrosomal cap? What is in the head? And all details. How is the tail? Okay. But they present in just one line in one sentence in each slide. Okay. Uh, Okay, anyhow, this head will have the cap called as acromosomal cap, acrosomal cap. Within this acrosomal cap is the enzyme called as the hyaluronidase. This hyaluronidase within the acrosomal cap will help in the breaking of these three barriers. What are the barriers? There are three barriers. One is called as the corona radiata. The first barrier is the corona. There are cells around the this oocyte. Oocyte is inside here. So this is the oocyte, and here is the oocyte. Around there are different many cells here around, arranged in radiating fashion. These are called as the corona radiata. They are called as corona radiata because they look like the radiation of a sun, radiations of the sun. They are arranged in lines, chains. Okay, they are radiating from the cell. So that's why they are called as the corona radiata. Then second structure is inside this yellowish structure called as the zona pellucida. Zona pellucida. And deep inside we can see the third membrane called as the vitelline membrane. These are the three within the wall of the this oocyte. So outside we have the corona radiata. Then inside this is the zona pellucida. This pinkish or reddish line around thick band, and inside we have the vitelline membrane. So these are the three barriers which will be broken down by the enzyme called a hyaluronidase, which is released from the acrosomal cap of the chromosome, the sperm. Okay, and that will break the three barriers around this oocyte. Oocyte is the female gamete. Okay. Okay, now these three barriers are broken down before fertilization, before penetration. Fertilization means penetration of the sperm. If you see here, these are the different sperms here, and one is getting penetrated. Okay, so before penetration, that is the spermatozoa has to undergo a process called as capacitation. So there are not only changes taking place within the oocyte, even there are changes taking place within the sperm. Okay, so it has to undergo capacitation followed by the acromosomal reaction to acquire this capacity. So only that sperm which will be able to have these two processes undergo these two processes called as capacitation and acromosomal reaction can have the capability to enter the oocyte. So what are these two, capacitation and acrosomal reaction? The first, capacitation, is the last stage of maturation of the spermatozoa. Even though the spermatozoa, the sperm, is maturing in the male reproductive system, but the final maturation of the sperm takes place in the female genital tract. Okay, so by the process called as capacitation. So capacitation nothing but the last stage of spermatozoa, the sperm in the female genital tract. So maturation of the sperm will take place actually in the female genital tract, and it requires about 7 hours. It requires around 7 hours to get this capacity. So the antigen coating on the surface of the sperms may involve to produce an immunological reaction between the fertilizing derived from the oocyte and the anti-fertilizing derived from the spermatozoa. So the antigen coating around on the on the surface of the sperm may help in the produce an immunological reaction between the fertilizing derived from the oocyte. So the oocyte fertilizing and the anti-fertilizing derived from the spermatozoa, though both combined together, and then it leads to something called as capacitation. Then there is second stage called as the acromosomal reaction. Acrosome. As I said, there is a acrosomal cap on the sperm. This is the sperm head. This is the tail of the sperm. On the top, there is something called as the acrosomal cap. Now there is some reaction going on there. Okay, just before approaching the oocyte, capacitated this is the capacitated sperm head establish multiple contacts between the plasma membrane and outer membrane of the acrosomal cap. If you see here, it is developing multiple contacts. Okay, so it is developing multiple contacts and there are some reactions going on. Okay, so it established multiple contacts between the plasma membrane and outer membrane of the acrosomal cap. So this is the plasma membrane here and the outer membrane of the acrosomal cap, and discharges acrosin and trypsin like chemical substances in succession to overcome barrier around the oocyte. So it is now, if you see here, it is releasing, okay, some acrosin as well as trypsin like substances. Okay, so by the hydrolytic enzymes. Hydrolytic means they are breaking down, lytic means breaking down enzymes. So they are breaking the barrier here. This process of multiple contacts as acromosomal reaction. So this multiple contacts and release of the hydrolytic enzymes, which will help in the breaking down of this barrier. This is called as the acrosomal reaction. So these are the two processes which will undergo. So before the sperm will be fertilized. So it has to undergo the capacitation, then it has to develop multiple contacts to have the acrosomal reaction. Then once it is successful, then the sperm head will enter. If you see here, the sperm head is entering. The tail remains outside, only the sperm head will enter. Now, the cells of the cumulus oophorus and the corona radiata are moved by the liberation of the hyaluronidase from the acrosomal cap. So now all the cells, if you see here, these are the differences. The sperm is approaching, it is coming in contact. So it is approach, now it is coming contact. Now there is capacitation as well as the acrosomal reaction, and then the sperm head will enter. During this process, there are cells here. These are called as the corona radiata, are moved by the liberation of the hyaluronidase from the acrosomal cap. So when it comes in contact, all the cells around that position will be moved. They will be moved. Okay. Then the next barrier is the zona pellucida. This is the zona pellucida. Zona pellucida, a thick striated membrane made up of 3 glycoproteins. So this is thick striated membrane made up of 3 glycoproteins ZP1, ZP2, and ZP3, synthesized by the oocyte. Oocyte is here, so it has synthesized this 3 glycoproteins, acts as the second barrier. Now, once the cells are moved and there is acrosomal reaction, now the second barrier is the zona pellucida, and now the sperm head has come in contact with the glycoproteins which are in the Z1, ZP1, ZP2, and ZP3. Now the sperm head binds to the ZP3 and ZP2 receptors and induces the acrosomal reaction to release the acrosin enzymes which digest the zona pellucida. So this sperm head will have the acrosomal reaction with ZP3 and ZP2 glycoproteins, and it helps in the digestion of the zona pellucida. Now the sperm head fuses with the vitelline membrane. Now inside, once it enters the zona pellucida, the third membrane is the vitelline, this thin membrane which you are seeing inside. This is called as the vitelline membrane, which acts as the third barrier. Two disintegrin peptides of sperm head open gate to form sperm to enter the oocyte cytoplasm. So this has to be disintegrated. This vitelline membrane has to be disintegrated before the sperm head enters the oocyte. So once the three barriers are broken down, now the sperm head enters. Once it enters, before it is entering, there is something called as calcium wave. So this is the acrosomal cap here. This is the acrosomal. This is the sperm head, this is the tail, and this is the sperm head, and this is the sperm head where the acrosomal cap is present, and it will release some enzymes which helps in calcium wave. So the calcium wave sets in throughout the oocyte cytoplasm, resulting in two important events. Now, once the sperm head has entered, so it leads to calcium wave within the oocyte, which leads to two changes within the oocyte. Okay. One is it triggers fusion of the cortical granules. If you see here, there are small granules within the vital membrane. There are small granules, round structures. These are called as the cortical granules in the periphery of the ooplasm, which release an enzyme that hydrolyzes ZP3 receptor on zona pellucida and prevents other sperms from binding. So it prevents polyspermia. So once there is calcium, once the sperm head has entered, then it leads to calcium wave, which will act on these small uh, cortical granules, and they all close this barrier on the zona pellucida, and they prevent other sperms from entering. So only one sperm can enter. One sperm enters, now there is a barrier formed. Okay, so there it prevents polyspermia. So poly means multiple, sperms means multiple sperms from fertilizing. It prevents other sperms from entering. Okay, and undergoes the acrosomal reaction. Granules also modify vitelline membrane and acts as the vitelline block. As I said, these granules will form a block here around, and it prevents any other sperm from entering. Once the sperm head has entered, so it leads to calcium wave, which will trigger these granules, and it makes a barrier around. Okay, completion of the second meiotic division. Okay, mature ovum with excursion of the second polar body is perivitelline space. So there will be exclusion of the second, release of the second polar body. So actually the oocyte will be still in the sperm will be the ovum will be still in the first meiotic division. You might have studied what is first meiotic division and second meiotic division during the gametogenesis. So this, as you know, it will be in the initial stages of the second meiotic division with the entry of the sperm head. Now the second meiotic division is completed, and there is release of the second polar body into the pre-vitelline space. Pre-vitelline space is a small space deep to the or outside the vitelline membrane. As you have seen, the vitelline membrane is just outside. Okay. Okay, now the cytoplasm of the mature ovum contains two pronuclei. As now the sperm has entered, so this is the sperm head, and this is the ovum head. Okay, which is the ovum female nucleus. So now it is called a female nucleus. This is called as a male nucleus. Both have entered, and the sperm head with the nuclear envelope and contains one haploid chromosome from male pronucleus and nucleus of the mature ovum from the female pronucleus. As I said, initially the sperm will have 23 chromosomes, and the male sperm will have 23 chromosomes. That is called as haploid. Okay. Now one haploid will come from female, one haploid of chromosomes will come together. Now it is diploid. Diploid means two halves, two halves. Okay. Now it becomes complete. So now it becomes 23 pairs. Actually, all our cells have 23 pairs of chromosomes already. But in case of sperm and worm, they will have haploid number, half number. When they combine, fuse, then it becomes diploid, the normal number, 23 pairs. The DNA replicates from 1n to 2n, as I said. Now 1n plus 1n becomes 2n. Okay. Now the DNA duplicates, then the pronuclear envelope disappears. The envelope around each of them will disappear, and the chromosomes each arrange in the equatorial plane. Now their chromosomes will all get arranged in the equatorial planes. Now if you can see here, there is a sperm coming, and this is the ovum head here. Okay. Now if you see here, this is the two-stage cell stage. We'll talk about that. The two heads, sorry, the two heads. One is the male nucleus, and the second is the female nucleus. Both have come together and they're getting fused. This is called as the fertilization. Okay, so here in this pictures, all the stages have been described. So here, if you see here, the sperm, one is the nucleus of the sperm and one is the nucleus of the ovum. Now they are arranged in the equatorial plane, and all the chromosomes half from the sperm and half from the female nucleus, they all are get arranged here in the this fashion, braiding fashion. So they form a spindle. Okay. Now, okay, so here you can see here the sperm head and the female head has come, and there is male and female pronucleus. Now here they are arranged. Okay, so this is the arrangement. So half from the male nucleus and half from the female nucleus. All get chromosomes get arranged. Now half of them will move to one side and half of the other, half of male and half of female will go to one side. Actually, first they duplicate. Before they go, they duplicate themselves, the chromosomes, and then half of them go to the one side and the half of them go to the other side. Now there is formation of two cells. Initially it was just one cell. This is one cell. Okay. Now there is beginning of something called as cleavage. So it starts dividing from one stage cell to multiple stage. So this is the process of division. So the chromosome initially they duplicate, they get arranged in the equatorial plane, and then half of them move to each side, and then there is division of the cell. So now one cell has become two cells. So this is how the fertilization will take place. So now coming to the what are the effects of fertilization? Why this fertilization should take place, and what are the effects? One is effect of fertilization is the completion of the second meiotic division. As I said, the the ovum when it is released, it is still in the initial stages of the second meiotic division. Once the sperm head enters, then there will be the second meiotic division has completion of the second meiotic division of the ovum. Okay, so if you see here, this is the ovary, and this is the fallopian tube, and here is the ovum coming and getting fertilized, and then we'll see what are the stages. Then restoration of the diploid number with the help of the entry of the sperm head and the female nucleus. Now there's restoration of diploid number. As I said, the sperm have half of the chromosome, the ovum will have half of the chromosome. With the combination of the two, it becomes restoration of the diploid. Restoration means bringing back to the normal. Okay, so it restores the diploid number, which is the normal number of chromosomes in the zygote. Now it is the fertilized egg is called as, it is not called as ovum now. Now it is called as zygote. When it is fertilized, it is called as zygote. Now the third is, what are the effects of fertilization? The third effect is determination of the chromosomal sex. Now at this stage, now we can already say the sex of the chromosome, even though we cannot make out with the ultrasound or anything, but the sex is already determined, whether this become male or female. As you know, all the X, the ovum will have X chromosome. All the X will, the chromosomes, the ovum will have X chromosome, and but the sperms, half of the sperms have X chromosome and half will have Y chromosome. Okay, so the ovum will have all X chromosome, but in case of sperms, half of the chromosome will have X and half will have Y. So depending on which type of chromosome has fertilized, if If you see here, this is the egg, the ovum which has X chromosome. Now the sperm with X chromosome is fertilizing this egg. Now it becomes XX, so that becomes female. On the other hand, the chromosome, the sex chromosome in the female is X, but the sex chromosome on the sperm will be Y. If the Y chromosome sperm will fertilize the egg, then it becomes XY, that is male. So it is the male sperm which will determine the sex of the chromosome. So at this stage, after fertilization, already there is already it is determined whether this fertilized egg will grow as male or female. So the determination of the chromosomal sex is already at the time of fertilization. Then the determination of polarity of the embryo, which pole will become head, which pole will become tail, that also is determined at this stage. Okay. Then the initiation of the cleavage division of the zygote. So as I said, cleavage means from one cell it becoming two cell become four cell, that is called as cleavage. So the beginning of the cleavage, initiation means beginning of the cleavage division of the zygote is already taking place at the fertilization. So these are the five things which will be the effects of fertilization, or the five effects of fertilization. One is there is completion of the second meiotic division. Second is the restoration of the diploid number of chromosome. Third is determination of the sex. Fourth is the polarity of the embryo, which will become head and tail, as well as the initiation of the cleavage of the zygote. All these will be determined at the time of fertilization. So these are the effects of the fertilization. Now coming to the cleavage. Cleavage. As I said, it is a process of repeated segmentation, not one time, two times, but it is repeated segmentation. Segmentation means breaking down of the zygote, that is the fertilized egg, within the zona pellucida. Still, you remember the zona pellucida? This covering here, red, pinkish, or the pinkish or the red covering. This is the zona pellucida. Here you saw the zona pellucida. This is the same zona pellucida. It will be still there, still present. This zona pellucida is very important. Remember, keep watching this zona pellucida. Okay, so this zona pellucida will be still there. So the fertilization, this division, cleavage is taking place within the zona pellucida, the outer covering. Okay, so it is a process. Cleavage is a process of repeated segmentation of the zygote within the zona pellucida by mitotic division. It is not meiosis, but mitotic division. So it means mitotic division means equal division of the cells in rapid succession. So it is repeated segmentation of the cells in very rapid succession. Okay, so this is called as cleavage. This is called as cleavage. So this cleavage, what will happen with this cleavage? It gives rise to smaller cells known as blastomeres. So the zygote, which is the fertilized egg, is the largest cell within the body. The cells normally they are not such big cells. Okay, so now it has to be reduced to bring it to normal size. So this cleavage will give rise to smaller cells, and they are called as blastomeres. So if you see here, this is the single cell. So with the two male nucleus and the female nucleus, with fertilization, now with cleavage, now it has broken down into two cells. Now the cells are smaller. With further division, they become still smaller. So one cell has become now two cells. So these are called as blastomeres. Okay, their cells are smaller as well as they're called as blastomeres. So if you see here, so there are group of cells here. They become large cells. Now with breaking down, they become smaller and smaller. Results in restoration of the normal size of the cells. As I said, the zygote is a large cell, so it has to be brought back to the smaller normal size. So this cleavage will result in restoration of the normal size, bringing back to the normal size of the cells by progressive reduction of the cytoplasm volume of the zygote. So as the between the cells, the cytoplasm will be divided. So as repeated division will take place, the cytoplasm become lesser and lesser. So the cell size will become smaller. So there are two types of cleavages. One is called as holoblastic and the second is called as the neuroblastic. Holoblastic means complete cell division. So this is called as holoblastic. One cell become as completely equally divided into two. So then the two cells equally divide into four. But neuroblastic is partial cell cleavage. Only a part of the cell will break and it will become the second cell. But here what is taking place is the holoblastic. What we have here in case of the zygote will be the holoblastic. So it is complete cell cleavage. So it means equal division of the cells. Okay. Now what happens? How they will divide? So the two cell stage from one cell to become this two cell, it takes about 1 day. Okay, after fertilization is called as the 0 day. At the time of fertilization is called as 0 day. So from there we start counting the days. Okay, so by the 1 day, in by 1 day it will take divide into two cells. One cell has become two cells. By the second day, the two cell stage become four cell stage. Okay, then by the third day it becomes 12 cell stage, and by the fourth day it becomes almost 16 cell stage, which is called as morula. We'll see that. Okay, so this is how it will divide. Okay, in the first day it will divide into two cells. By the second day, 4 cells. 3 days, 12 cells, and by the fourth day it becomes 16 cell stage. So now here you can see here. So this is the whole process shown. So here is the ovum, really the ovary releasing the ovum, and now it is fertilized here by the sperms. If you see here, so this is the ovum. Now it is fertilized by the sperms. One sperm had has entered. Now there is male nucleus and female nucleus, and this is called as the zygote. All other sperms are outside. Now it starts dividing into two cell stage, then it becomes four cell cell. See, at the same time when it is dividing, so the fertilization takes place in the ampulla. Now with the division, it is moving towards the uterine cavity. If you see here, it is moving towards the uterine cavity. Okay, as at the same time when it is dividing, it is also going into the uterine cavity. So the one cell stage has become two cell has become four has become eight has become the 16. Okay, by the 16 cell stage it already reached the uterine cavity. Okay, now it changes into something else. We'll talk about that. So the morula, as I said, it is the 16 cell stage of the zygote, as we saw here, 8, and then it becomes 16 cell stage. So this 16 cell stage of the zygote, this is called as the morula. It is called as morula because it looks like a mulberry. Mulberry. That's what it's called a morula. And it takes, as we saw, it takes almost 3 to 4 days. Okay, for this stage to reach. Now all the cells of the morula are similar in size and structure. As you can see here, all the cells are in equal stages, and around that this is the zona pellucida. Remember, as I said, zona pellucida is very important. Keep watching. Still the zona pellucida is there, and this cells are 16 cell stage, which is called a morula, and all the cells are equal cells. Okay. Now, the group of cells in the center of the morula known as the inner cell mass, which gives development to the embryo proper. Now, the cells divide themselves into outer cells and the inner cells. Initially, they will be all equal. Now, they themselves distribute themselves into inner cells and the outer cells. Outer cells, these are outer cells, outer layer of cells and inner cell mass. So this inner cells mass will develop itself into the embryo proper. So from this inner cell mass will be the embryo proper. Embryo means embryo will develop from this inner cell mass. So what happens to the outer cells? The cells at the periphery are called as the outer cell mass. These are outer cell mass, which have protective and nutritive covering of the embryo. So this will play a role in protecting the embryo, outer cell mass, as well as they form a covering and they provide nutrition to the embryo, so which will later form the placenta. which will form the placenta. We'll see that stage. Okay. So the cells of the marula are still covered by the xenophilus. As I said, so there is one cell to becoming two cell, two becoming four, four becomes eight, and four become eight becomes 16, and still the marula is covering. Now the cells of the marula are divided into two types: inner and outer cell mass. Inner cell mass will form the embryo, and outer cell mass will form the placenta. Okay, still it is covered by the zona pellucida. This is the zona pellucida. Okay, now it changes into something called as blastocyst. Blastocyst. So morula changes into blastocyst. This is the morula. Okay, with the inner cell mass and outer cell mass. Now it changes into something called as the blastocyst. As the cells of morula continue to divide within the uterine cavity, fluid from the uterine lumen called as uterine milk passes through the zona pellucida and outer cell mass of the morula and appear in the inner cell space between the inner cell mass and outer cell mass. Means, so as you see here, when it reaches the uterine cavity, it is already morula stage. Now there is some fluid in the uterine cavity, starts entering into the morula. It starts entering into the morula. Now there is creation of a space within the morula. So this is the morula. Now there is fluid entering here. This is called as blastocele. Blastocele with fluid inside. And this embryo proper inner cell mass is pushed to one side. Okay. So the uterine cavity, so the fluid in the uterine cavity, which is called a uterine milk, enters into the morula and creates a cavity called as blastocyst or the blastocystic cavity or blastocele, and the inner cell mass is pushed to one side. Okay. Okay. Continued accumulation of the fluid causes coalesce of the inner cell spaces and converts morula into large fluid-filled cavity called as the blastocele. So as I said, this cavity is formed, is called as blastocele. This is the blastocele. The cells of the outer cell mass are flattened, form the wall, and now called as the trophoblast. Now the cells of the outer cell mass, which we saw here, these are the outer cell mass. Now they changes themselves, they become flat, and now they are called as trophoblast. Very important structures. Trophoblast. This is the trophoblast which is formed around. The cells of the inner cell mass are called as embryoblasts. So the cell's inner mass, cell mass, which we said, which will develop into the embryo proper. Now it is called as the embryoblast. So the outer cell mass, this is called as the trophoblast, and inner cell mass, now it is called as the embryoblast, which is attached to one pole. So it is attached to one pole, and the remaining part is filled with the cavity called as blastocele, and this whole structure is called as blastocyst. So this is how it looks like. This is the real picture of the blastocyst taken under the electron microscope. Okay. So here is the blastocele, and this is the inner cell mass, which is called as embryoblast, and outside we have the trophoblastic cells. Okay. Now the trophoblast covering embryonic pole of the blastocyst is known as the polar trophoblast. Now it goes to one side of the pole. Now this is called as the polar trophoblast, where the embryo is attached. That is called as the polar. This trophoblast. Okay, these are attached to one side, so that part is called as the polar trophoblast. Okay, and the rest are called as the mural trophoblast. So the rest of these trophoblasts, so these are called as trophoblasts. Now they are called as the mural trophoblast because the embryo is not attached to this wall; it is free. But one side it is attached, where the embryo is attached, that is called as the polar trophoblast. And the remaining part is called as the mural trophoblast. The blastocyst is formed between the fourth and fifth day after fertilization. As we saw, by the fourth day it will be the morula formation, and by the fifth day it is the blastocyst formation. So you know the stages, how it has changed. It has staged from the first one single cell stage to two cell, then 4 cell, then 8 cell, then 16 cell. This is called as morula. Now the cells in the morula changes into outer cell mass and inner cell mass, and there is formation of a cavity. Now it is called as the blastocyst, and here this blastocyst will appear by the fifth day, 4 to 5 days after fertilization. As I said, fertilization will be counted as the 0 day, and from that day we start counting the days. So by the 5th day, there is formation of the blastocyst. Now the blastocysts have 107 cells. Okay, by this stage already it has divided into 107. So from one cell to 107 say, okay, so that's why we said it is rapid and successive, very rapid division. By the 5th day already there are 107 cells of the blastocysts. Out of this, they are divided into three types of cells. 8 cells from the embryo from out of 107 cells, 8 cells from the embryoblast, and 30 cells from the polar tropoblast. Polar tropoblast, where the embryoblast is attached, that is called a polar tropoblast, and the remaining 69 cells from the mural trophoblast. These are the mural trophoblasts. Okay, so the same cell stage you can see here going on. So as it is breaking down into multiple cells, it is going towards the uterine cavity. You should remember. So one cell stage has become 2, 2 becomes 4, 4 has become 8, 8 has become 16, and now there is formation of the blastocyst. So it has reached the uterine cavity. Now it is flowing in the uterine cavity. Now there is something called as implantation. Implantation, this is the last thing today we'll study, that is called as implantation. So now if you see here, implantation means what? Getting attached to the uterine wall. So this is called as the implantation. Okay, so what is implantation? Implantation is embedding of the blastocyst in the endometrium of the uterus. What do you mean by endometrium? This is the wall of the uterus. So the wall of the uterus are three layers. Outside is called a perimetrium, this is called a muscles, myometrium, and this is called as the endometrium. So the inner wall of the uterus is called as endometrium. So now the endometrium is well ready for the implantation. This is called as implantation. So if you see here, this whole of the blastocyst has come and get attached. Okay, this is called as implantation. Implantation is embedding of the blastocyst in the endometrium uterus approximately a week after fertilization. Week means almost 6th or the 7th day of fertilization. So it gets implanted within the wall of the uterus. So if you see here, so this is the wall of the uterus in different stages of the menstrual cycle. You might have already studied this menstrual cycle and how the endometrium starts growing. Okay, and how it is ready during the progestational stage or the secretory phase, and now it is ready for the to receive the fertilized egg. Okay, that is the blastocyst. Now it has get implanted within the wall. If you see here, it get entered. Okay, so for this implantation to takes place, the zona pellucida should disappear. Zona pellucida disappear just before implantation. So for till now, the implantation was not taking place anywhere till here because the zona pellucida, which was the outer covering, was still there. Now, if you see here, now this is called as the hatching of the blastocyst. This is the blastocyst. Now the blastocyst will come out of this zona pellucida. So the zona pellucida has to disappear before it gets implanted. So till now it was not implanting. So because it was there was the zona pellucida. Now we can understand the importance of zona pellucida. If it disappears early, then there can be abnormal implantation within the fallopian tube itself. Okay, so we'll study the abnormal implantation where you see that it is get implanted within the wall of the fallopian tubes instead of here. Why do it gets implanted in the uterine cavity? Because the zona pellucida disappear now. If it gets disappeared early, so it will implant here itself within the fallopian tubes. Okay, so this is the importance of the zona pellucida. So by the 7th day, zona pellucida will disappear, which helps in the implantation because the trophoblastic cells are very much ready to get implanted. So it was prevented by the zona pellucida. Now it has disappeared, so it can easily get implanted. At the time of implantation, the uterus is in the secretory phase. As I said, it will be the secretory phase, that is the progestational phase, during which the time uterine gland, these are called as the uterine glands, and the arteries, these are the arteries, become coiled. As you can see here, there is high coiling of the uterine glands as well as the arteries, and the tissue becomes succulent, that is, it becomes thick and ready for the implantation. So three distinct layers can be recognized in the endometrium. You might have already studied the three layers within the endometrium. That is called as the stratum compactum outside. Then this big layer, this is called as stratum spongiosum, and the lowermost layer called as the stratum basale. These are the three layers, and this get implanted into the compactum and the spongiosum. So the endometrium of the uterus helps in the implantation and contributes in the formation of the placenta. Now, as we saw, this is the endometrium. So it helps in the implantation of the if you see here, so it is getting implanted within the wall. Okay, so it helps in the implantation of the blastocyst, as well as also helps in the formation of placenta. We'll see this development of placenta later. Okay, usually the human blastocyst implants in the endometrium near the fundus. You know the fundus of the uterus. The fundus of the uterus is on the near the top. Okay, where the fallopian tubes open on either side. Above that, whatever region is there, that is the fundus. So usually the implantation will take place in the fundus along the anterior or the posterior wall. Usually near the fundus in the posterior wall of the uterus, where it becomes embedded between the opening of the glands. But during multiple pregnancies, the implantation will be much more below. Again, this will lead to a lot of complications. We'll study all these things later. So, usually it should get implanted in the upper part. Okay, if it gets implanted in the lower part, then it leads to a lot of complications. Okay, which is called as the placenta previa and other things, which we'll study later. Okay. So as I was telling about the importance of the zona pellucida, we'll just identify what are the different abnormalities of implantation. Okay, one is the commonest, one of the common abnormal implantation will be within the abdominal cavity. Usually, instead of fertilized egg going into the uterine cavity, it will move out and get implanted within the abdominal cavity. It is rare, almost 1.4% usually in the pouch of Douglas. There is a pouch called as pouch of Douglas. But the most commonest will be the ampular region of fallopian tubes. If there is implantation abnormal, usually it will be the ampullary part. What is the ampullary part famous for? It is the site where the fertilization takes place. So once there is fertilization takes place, the zona pellucida, if it disappears immediately, then it gets implanted there itself. So almost 80% of the abnormal implantation will be taking place in the ampullary region of the fallopian tube. So this is the commonest. Then it can also get implanted in other parts like tubal implantation in the remaining part in the tube, in the isthmus or if it implanted slightly later but earlier than before it reaches into the uterine cavity, it can implanted in the tube, other parts of tube like the isthmus as well as the intramural part. This is almost 12%. Okay, this is the second commonest. This is the commonest, and this is the second commonest. Then it can also get implanted. This is the third type, that is called as the interstitial implantation. Interstitial implantation. So where it gets implanted within the interstitial wall. Sometimes there can be internal os. Internal os is a part of the cervix. This is the cervix. There is internal os and the external os. So sometimes it goes to the cervix instead of, as I said, this is the fundus. It should be implanted on the fundus or the posterior wall, but sometimes it goes into the cervix and get implanted. Okay, which results in, I was telling, which leads to placenta previa, almost 2.2%. And sometimes it get implanted in the ovary. Instead of going here, the fertilized egg come here and get implanted in the ovary. This is also 0.2%. But the most commonest will be the fallopian tube, especially in the ampullary part. And the other type of the fallopian tube is in the other parts, that is 12%. Okay, so these are some of the sites of abnormal implantation. So this is all about this lecture called as the fertilization, as well as the stages, different stages of fertilization, as well as the cleavage. Okay, as well as even the implantation. So if you have any doubts regarding this, you can ask me.
Doctor,
Yeah.
Doctor, I can't regress attendance in the model.
If you are late, you should come early. It will be just for 15 minutes. Don't contact me later that we are late or something we missed. This is not fair. because we give first 15 minutes to mark your attendance. 15 minutes is too much for you. Okay, but still if you don't mark it, I don't know what should we do. Okay. Any questions on this topic?
No, thank you, doctor.
Okay, go back home, read, because reading is very important, because the next stages will be related to this. If you don't understand this concept, you cannot understand the future lectures. So go back and read it. If you don't understand, you can contact me and you can clarify your questions. Okay. Okay, read it before you come to the next lecture, because it is very important to understand this lecture, because all these lectures are related to each other. If you don't understand the first lecture, you cannot understand the second. If you don't understand the second, you cannot go to the third. So try to be read regularly and try to understand. Okay. Any other questions?